The eye is I think, one of the most fascinating organs in the body. And I'm going to leave it to the neurophysiologists and the ophthalmologists to explain all the detailed physiology and also some of the anatomy of the eye. But in this lecture, I'm going to describe all the major components of the eye. I'm going to outline their histological structure and then indicate what functions each of those structures have in the whole function of the eye, giving us vision. At the end of the lecture, you need to understand, or at least identify, the structures that I'm going to talk about. You need to understand the role of the cornea and the lens in focusing light on the retina, and the role of the iris in being a diaphragm and restricting or opening the amount of light going through the pupil to the retina. And I'm going to describe the muscles that change the dimensions of the pupil. And you need to understand the function of those muscles as well. I'm also going to describe the production of the aqueous humour by the epithelium of the ciliary processes, and how that aqueous humour circulates through the posterior and anterior chambers of the eye, and finally how it's absorbed into the canal of Schlemm. So it's important that you understand all those processes. You need to understand the role of the retina, in particular, to be able to identify the rods and the cones and understand a little bit about their functions. And also, I want you to be able to understand the cause of presbyopia, which is a condition of aging of the lens, and then also what constitutes the blind spot. Well, as I said at the start of this lecture, the eye is probably the most fascinating organ in the body. The cornea and the lens focus light on the retina. The illumination is adjusted by the iris. The rods and cones are the receptors, the photoreceptors that send impulses to the brain to enable us to see, to have vision. And then extraocular muscles, which I won't describe in this lecture. I'll leave that to the anatomists. The extraocular muscles actually move the eye, and they're coordinated by very precise impulses coming from the brain. But as I said, I'll leave that to the anatomist to describe for you. Let me just outline the general structure of the eye. On this diagram, on the right-hand side, it outlines most of the structures that I'm going to describe histologically. But I want to just remind you that on the left-hand side, the embryologists will explain to you in embryology lectures that the eye is actually derived from 3 different sources. It's developed from those components I've listed there on the left-hand side. So we're now going to look at each of those structures in turn. I'm going to start with the cornea, the front of the eye, the anterior part of the eye, and go through the structures in sequence, from the anterior structure of the cornea and then to the retina. First of all, let me just outline the structures that are actually produced by the 3 different sources. On the left-hand side, the outer layer, or tunic, consists of the sclera and the cornea. The cornea is the clear, transparent anterior portion of the eye. The sclera is the white portion. It's the very fibrous, tough, fibrous outside of the eye that the extraocular muscles insert on. The sclera in young children is quite thin and has a bluish tinge to it. As we age, it has a yellow appearance to it, and it thickens. The next layer, the next inner layer, consists of the choroid, the ciliary body, the stroma of the ciliary body, and the iris. This is termed the uvea. The choroid is the vascular supply to all these structures I'm going to describe. The choroid's very important because it provides support and blood vessel supply of nutrients to the retina, which is avascular. And then the most internal layer, the inner tunic, is the retina. And the retina consists of two parts. The neural part, which extends towards the front of the eye and ceases to be the neural part at the ora serrata. And then it extends to cover the ciliary body, ciliary processes, and the iris as a double epithelial layer, which I'll describe later on. The ganglion cells project their axons from the retina into the optic nerve. And notice in the diagram on the right-hand side that the optic nerve is actually covered by meninges, the dura, arachnoid and pia. So we shouldn't call it really an optic nerve. We should call it the optic tract because it's actually brain tissue. Okay, let me start off, as I said, at the front of the eye at the cornea. The cornea is shown in low magnification on the left-hand side, and then increasing magnification in the middle and also on the right-hand side. The cornea consists of 2 cell layers and 3 other non-cellular layers. And the thickest component is the stroma, labelled here. That corneal stroma is a transparent part of our eye, the cornea. It provides transparency. And it does so because when you look at the fine structure of the stroma, it consists of about 60 layers of collagen fibrils. And those layers are arranged in 90-degree or perpendicular to each other, layer on layer on layer. And they're held together by glycoproteins and collagen type 5. And those glycoproteins, those proteoglycans rather, and collagen 5 actually hold all these bundles of collagen fibrils in their arrays and create this transparency. The corneal epithelium, the surface epithelium, is a stratified squamous epithelium, as you see in this slide. The very surface cells, the squamous cells you see at the surface, have very fine microvillous projections, and those projections help to maintain tears on the surface of the eye. And those tears protect the eye, and they also give some ultraviolet radiation protection from those cells within the cornea. Those epithelial cells don't have melanin in them like you see on the skin surface to protect from excessive ultraviolet radiation. But the nuclei have in them ferritin, which gives that protection to the nuclei from excessive ultraviolet radiation. The corneal epithelium can replace itself very rapidly, but it doesn't replace itself from cells that are immediately at the base of the epithelium there. They replace themselves by a stem cell niche or population of cells in the periphery, right next to the junction of the cornea with the conjunctiva. And that's to protect the cornea from conjunctival epithelium migrating its way across and being part of the cornea. The epithelium, the outer epithelium, is supported by Bowman's membrane. This membrane is a very thick membrane. It's a big, thick basement membrane. And once it's damaged, it can't regenerate. On the other side, the inner aspect of the cornea facing the anterior chamber is another basement membrane. It's a thinner basement membrane. This can repair itself on a regular basis if required. And then the surface cells, the inner surface of the cornea, is lined by a cuboidal epithelium, the corneal endothelium it's called. Unusual name, endothelium, really refers to the lining of a blood vessel. But here, there's a special exception for the cornea. Those cells are very, very metabolically active. They provide nutrition and support for all the fibres that make up the cornea. The cornea is avascular. It's separated from the body, the vascular system of the body, and therefore it can be used as as a donation, corneal transplants. It doesn't suffer from any immunological attack, and that's why it can be easily donated and transferred from one person to another for a person who is in need of a corneal transplant. The cornea then becomes continuous with the sclera of the eye and the junction of the epithelium with the conjunctiva. I'm going to come back to this image later on because I'm going to describe the canal of Schlemm, which is a very small network canal you can see at the very corner of this image, but I won't bring it to your attention yet. Now let's move back from the cornea and look at the iris. It's the diaphragm that allows certain amounts of light through the pupil and through the lens. It guards us against excessive illumination, and it opens up and allows more light to come through the lens or through the pupil when we're in dim light. So it needs to be a structure that can contract and open or dilate, contract or constrict. If you look at the right-hand side, you can see on the most posterior aspect, there is this heavily pigmented layer. In fact, there's two layers of cells there. They are part of the extension or the continuation of the non-neural part of the retina. Double layer of pigmented cells. On the right-hand side, the anterior border, it's a rather rough border, and that can be seen with the ophthalmoscope when examining the eye. Now, within the stroma of the iris, there are also melanocytes. And when light passes through and reflects from that posterior pigmented layer, if there's not too many melanocytes in that stroma, the iris has a bluish colour. But as more melanocytes are present in that stroma in various individuals, the colour of the iris changes from a greyish blue to a green to finally a brown as more and more melanocytes are embedded in that stroma. The iris sits on the lens, and as I said, it changes its dimensions to allow more or less light going through the lens depending on the level of illumination. And the lens and the iris are associated with the ciliary body, which I'll describe in a moment. When you look at the iris in higher magnification, as we'll do in a moment, this is just to remind you of where the iris is, we can see that the iris has within it two different sorts of muscles shown on this particular diagram. In the diagram, you can see the very tip of the iris. On the right-hand side, there's a little opening where the lens is, that's the pupil. And I bring your attention to two muscle layers, or they're really a myoepithelial layer and a smooth muscle layer. The dilator pupillae consists of myoepithelial cells, and they run like the spokes of a wheel. They run radially out from the inner part or the edge of the iris to the external part, as I said, like the spokes of the wheel. And if they contract, then the pupil's going to dilate. If the spokes of your wheels contract, then they open up a space where the hub of the wheel is. Conversely, at the very tip, there is a sphincter pupillae, and that runs in a circular pattern around the iris. If that contracts, then it restricts or reduces the diameter of the pupil and therefore restricts the amount of light going through. And as you can see explained here, they're innervated by two separate components of the autonomic nervous system. Let me now move from the iris to the ciliary body, shown here in the diagram. It's got a number of components which I would like to explain to you. First of all, the ciliary muscle is the bulk of what you see on the top left-hand side of this picture. Ciliary muscle is very important because when it contracts or relaxes, it affects the dimensions of the lens, because the ciliary processes which you see there, they have an epithelium on them. There's two epithelial layers there, and again they're extensions of the non-neural part of the retina. Those epithelial cells closest to the posterior chamber or the vitreous actually make these little fibres, and those fibres extend to the lens. And as we'll see in a moment, when the ciliary muscles contract or relax, it changes the tension on those fibres extending to the lens, and therefore the shape of the lens. So the ciliary processes are very important for that. And also the epithelial cells produce the aqueous humour that I'll explain in a moment. So let's just go back and look at the image on the right-hand side. Let's look very carefully. You can see some of these zonular fibres extending from the ciliary processes. They're very hard to see in sections because often when you section through the eye, you just don't happen to be lucky enough to get these zonular fibres. But some are shown here if you look very, very faintly. And the top of the lens is shown up in the top left-hand side of this particular image. And there's the ciliary muscle there in the corner. And on the right-hand side of that, the dark green structure is going to be the sclera. You're looking right at the junction between the cornea and the sclera in an area we call the limbus. Let's just go back a step before we continue with those zonular fibres and the lens and have a closer look at this double epithelium that lines the ciliary processes. You can see two epithelia here. Adjacent or facing the posterior chamber or the vitreous is the non-pigmented ciliary epithelial cells. They're cuboidal cells. And then below them are the pigment ciliary epithelial cells. And then below those cells is going to be the stroma of the ciliary body. And again, let me remind you that this double epithelial layer is the non-neural extension of the retina. Back in the posterior part of the retina, of course, there are going to be the photoreceptor cells and also this pigmented epithelium. Right between the two cell layers is the ciliary channel, which is an important collection site for when the process of these cells are making the aqueous humour. And if you look very, very carefully at the top part of the epithelium, the non-pigment epithelium, you can see a very faint line running across the surface of those cells. That happens to be the basal lamina, the basement membrane of this epithelial layer. The basement epithelium of the pigmented epithelium is adjacent to the stroma. So really that ciliary channel you see labelled there has both the apical surfaces of the non-pigmented epithelium and also the apical surfaces of the pigmented epithelial cells. And we're going to see in a moment that that basal lamina on the surface, on the non-pigmented layer, are going to be the component that anchors the zonular fibres that extend then to the equator of the lens. Here is a diagram explaining that double epithelium that I indicated to you in the previous slide. I actually like this diagram. I like the colour in it. So congratulations to the artist. But focus on the bottom part of the diagram. You can see a fenestrated capillary. That's the stroma component of the ciliary processes in the ciliary body. And just above that is the pigmented epithelial cell, then the ciliary channel, and then the non-pigmented epithelial cells. And then you can see the basal lamina on the non-pigmented ciliary epithelial cells, and extending from that basal lamina are the zonular fibres that are going to go to the equator of the lens. And then above that, it just tells you really the components of the aqueous humour. They are produced, those components, by the cells, the epithelial cells. particularly the non-pigmented cells, they're very, very busy. They're very busily processing various components coming up from those fenestrated capillaries. They're actively processing materials. So they have lots of sodium and potassium pumps, etc., and a very busy epithelium that makes up this layer. Busy because not only are they producing the aqueous humour, but they're also producing those zonular fibres as well. So they're busy cells. And that aqueous humour is going to circulate through the anterior and posterior chambers of the eye. Now I can show you the canal of Schlemm, which sits in the limbus that I pointed out earlier. There's the corneal epithelium coming around to join the conjunctiva, and the cornea is continuous with the sclera, and there's the canal of Schlemm on the left-hand side in low magnification, and on the right-hand side in the centre at high magnification. This is a network that's going to allow that aqueous humour to drain into it and then find its way into the venous system. Here is a diagram explaining that. The pictures on the left-hand side and the diagram just follow the sequence. 1, the epithelial cells produce that aqueous humour. It circulates from the posterior chamber through the little valve created by the iris into the anterior chamber and then into the canal of Schlemm and then to the venous system. That's constantly being produced, and it maintains a certain pressure in the anterior and posterior chamber of the eye. If for some reason there's a blockage in that canal or overproduction, then the pressure increases. And that's a condition that we call glaucoma. And it's quite a common eye disease that needs to be treated. The vitreous cavity, shown in the very bottom part of the diagram, is a gel-like component. It's a large component. Its job, being a gel, is to maintain the shape of the eye, but it's also a shock absorber. When you do sudden eye movements, the vitreous protects the retina, the very sensitive retina, from vibrations caused by those rapid eye movements. You can see the lens there, and you can see the zonular fibres extending from the epithelium I described earlier to the equator of the lens. Again, let me remind you that those lens fibres, or zonular fibres, are produced by the epithelial cells that produce the aqueous humour. So let's look at the lens now in more detail. On the left-hand diagram, it's a large picture, you can see the zonular fibres extending from the ciliary epithelium of the ciliary processes, and then you see the structure of the lens. It's got a capsule all the way around it, a clear, transparent capsule. And then underneath that capsule is an anterior epithelium, the lens epithelium. It's only on the anterior surface. The posterior surface doesn't have that epithelium. And those anterior lens epithelial cells produce the lens fibres. And those lens fibres, if you look on the right-hand picture now, you see at the equator, that epithelium produces the lens fibres, and then those fibres move towards the centre of the lens. And their nuclei, as they move towards the centre, start to disappear and finally end up as what we call ghost nuclei. They're just little spaces that are finally filled up. Now, these lens fibres could be anything up to 8 to 10 millimetres in length. They're about 8 to 10 microns in width, and they're packed all together. And as they move towards the nucleus, after being constantly produced at the equator, they tend to fuse together, and it's very hard to separate or see the different fibres lined up. And as they move towards the centre, they also accumulate the protein crystallins. These lens fibres are produced regularly. They consist of type 4 collagen, and it's very elastic. And that elasticity changes with age. It gets reduced as we get older. When you're in the 4th decade of your life, suddenly the elasticity changes so that the lens can no longer accommodate, change its shape as well as what it did in the younger person. That's called presbyopia, and therefore the person normally has to wear glasses to accommodate for that, to assist the lens, to focus the light rays on the retina. And you can see here, again, the zonular fibres extending from the ciliary processes to the lens. And this summarises what happens when the ciliary fibres are stretched or put tension on or they relax, caused by the ciliary muscle. On the left-hand side, when the ciliary muscle contracts, the ciliary body squeezes up and moves closer towards the lens. So the zonular fibres become more relaxed, and so the lens will fatten out. It will become wider. Conversely, when the ciliary muscle is relaxed, then the ciliary body moves away from the lens and the fibres become stretched or tense, and therefore the lens gets thinner, changes shape. And this is how the lens, therefore, can focus light from distant objects or close objects regularly onto the surface of the retina, the process of accommodation. Let's now move back and look at the choroid. I mentioned earlier it supports the retina, shown in this slide. It's a pigmented layer. It's got pigment within the stroma, but it's dominated by capillaries, by blood vessels. And these blood vessels also pass on to the ciliary body, the ciliary processes. And you can see those blood vessels when you examine the internal structure of the eye with the ophthalmoscope. That pigment and the pigmented epithelium of the retina help to stop scattering of light around the inside of the eye chamber and therefore glare. On the double pigmented layer on the posterior aspect of the iris, that absorbs light coming in. And as I said, if it's reflected back, it can determine the colour of your iris depending on the population of melanocytes in the stroma. But the choroid essentially is a vascular layer. It's important because, as I mentioned earlier in the lecture, it provides nutrition to the avascular retina, particularly the very important ganglion cells, neural cells there, and also the photoreceptors. And below you can see it's continuous then with the outer fibrous coat of the eye, the sclera. On this slide, you can see on the right-hand side, the sclera coloured green, and then this choriocapillary layer. You can just see little red blood vessels in this capillary layer, and it's separated from the pigmented epithelium and the retina by this membrane. It's a very thin, faint membrane that runs between the pigmented epithelium and the choroid. It forms part of the barrier, the blood-retinal barrier. Well, let's look at the retina. And goodness me, look how complicated it is. On the right-hand side is a picture taken from a light microscope of the retina and all its layers, supported by the pigmented epithelium and the choroid. And that coincides with a diagram in the centre, and then a picture in the centre, I should say, and a diagram on the left-hand side. I'm not going to go through all these cells and all the process of vision. As I said earlier, it's the role I will leave to the neurophysiologists and the ophthalmologists. But I'll just point out a couple of things. On the diagram, focus on the rods and the cones. They're shaped differently. They have outer segments that are in contact with the retinal pigment epithelium. Those rods and cones are in various concentrations around the retina. Cones are specialised for colour vision and rods for vision in dim light and peripheral vision. There is a part of the eye, the macula, and in the macula, which is about 4 or 5 millimetres in diameter, There is a central part of that macula called the fovea centralis. It's about 1.5, maybe 2 millimetres in diameter. And within that, the central part of that is the foveola. That's about 200 microns in diameter. That's fully concentrated by cones only. And that's where our focus of light rays is perceived to be the strongest. It's where we can perceive the most detail of structures, and it's where colour is perceived. It's a high concentration, as I said, of cones. Those cones are quite large. They can be 80 to 60 microns in height. The rod's a little bit smaller. And as I said, those cones are concentrated in that foveola. There's about 4,000 of them there. And they occupy that space. And also, if you look at this diagram, at the foveola, the central spot, the retina is slightly tilted on an angle so that the light actually hits the photoreceptor cells without passing through all the layers of the ganglion cells and neural cells supporting those cells, those photoreceptors. In that area, in the foveola, there are 4,000 cones, as I pointed out, and the ratio of cones to the ganglion cells is 1:1, whereas elsewhere it's much higher than that. There are, in fact, about 120 million rods and only about 5 to 7 million cones. So about 125 to 127 million photoreceptors. There's only 1 million ganglion cells. So each ganglion cell, therefore, sends impulses from a combination of these photoreceptors. I want to just turn your attention now to the pigment epithelium. That pigment epithelium is a very important structure. It has a number of different jobs to do. The outer segments of the rods and cones consist of discs containing the visual pigment, which are shed continually. And one job of these retinal pigment epithelial cells is to phagocytose those discs being shed, and then to also re-photosensitize the visual pigment that has been dissociated in contact with light. They also form a very important blood-retinal barrier. So they're very, very important cells. And as I said, the pigment helps to stop light from being reflected back into the eye and therefore creating glare. Let me just point out the structure of the optic nerve. In brackets there I've put tract for the reason I explained earlier in the lecture. On the left-hand side you can see the optic nerve with a very characteristic central artery in the middle. And the purple stained components you see there are all the axons, and they're supporting cells. Those axons are projecting from the ganglion cells in the retina at the optic disc, which doesn't have any photoreceptors in it, so that's our blind spot. On the outside of that image, that section, you can see a dense connective tissue layer. That's the dura, one of the outer meninges of the brain, protecting the brain and spinal cord. Then you see an artificial space, a white space, and then a thin line. Normally that thin line is up against the dura, and you have a space, that thin line is the arachnoid layer. And underneath that space is real. That's the subarachnoid space. And on the right-hand side, at higher magnification, you then have the pia, directly in contact with the surface of the ganglion axons, unmyelinated nerve fibres. And that's the pia component, and that actually extends into the bundles of unmyelinated axons and supports them. So the optic nerve is surrounded by meninges, not the epi, peri, an endomysium you find around peripheral nerves, which is why I keep saying it should be called a nerve tract rather than just a straight peripheral nerve. So optic tract is a better name for this structure. The supporting cells, you can just see little nuclei in there. They're going to be glial cells. The eyelid is a structure that protects the eye. We saw the conjunctival surface, a continuation of the corneum, very early on. Remember, the junction is protected by that niche of stem cells that provide additional cells to the corneum when they're lost, and it prevents the cells lining the conjunctiva from being part of that epithelium. The conjunctiva is it's a wet mucous surface. There are a number of different glands associated with the eyelid. The meibomian glands secrete large amounts of secretions. They secrete an oily component, and that oily component mixes with the tears that are secreted by the lacrimal gland and also small lacrimal glands in the eyelid. And that oil prevents the very rapid evaporation of the tears and therefore maintains the protection of our eye, particularly the corneum. The orbicularis oculi is in the centre of the eyelid. That's one of the very small muscles of facial expression. And then there's a very dense piece of connective tissue running through the eyelid called the tarsal plate. That gives the eyelid a certain structural rigidity or support. The lacrimal gland is just like any other compound gland. I described the structure of compound glands in another lecture. But as you see on the left-hand side, under very low magnification, you can see clusters of glandular units called lobules. And within those lobules, on the right-hand side, you can see secretory acini, little grape or berry-shaped cells. They're serous secreting. They secrete a watery fluid with some material in it that helps to be antimicrobial. In other words, it stops bacteria and stuff, other material from colonizing on the surface of the eye. They are surrounded in the interstitial components by plasma cells that secrete antibodies through those tears again onto the eye to protect it. And the rather large circular structure you see on the right-hand side is a duct lined by an almost columnar epithelium. But I won't say any more about the lacrimal gland because, as I mentioned before, it's covered in lectures I've given in this course on the exocrine glands. In summary then, make sure you understand now the structure of the cornea and the lens and how those two major components then focus the light on the retina. Understand the macula lutea. It's a yellow pigmented area of about 5.5 millimeters in diameter, I mentioned, and the fovea centralis with the very central fovea. That is where the light is focused when we want to have detail, discrimination of detail, and also very fine colour vision. It's where there's the concentration of cones only. And then there's the optic nerve, containing all the ganglion cells, axons projecting back to the brain. There's the retina that I've described, all the photoreceptors there, and the very complex supporting cells and other neuroglial cells, neurons, and conducting neurons within that retina. And then there's the production of the aqueous humour by the ciliary epithelium and the production of the zonular fibres that extend to the lens. And then the importance of the ciliary muscle, when it contracts and relaxes, it changes the shape of the lens and therefore allows us to focus distant objects and near objects on the retina. So I hope you now understand a bit more about the eye, particularly the histological structures of the eye, and thank you very much for listening to this lecture.